Hello everyone, welcome to One Page Biology. In this video tutorial, we are going to discuss about how the membrane phospholipids are formed. So, in the earlier video, we saw how the formation of fatty acids take place. Now, let us understand how the particular phospholipid is formed. So, as we know that phospholipid can be monoacylglycerol or diacylglycerol or triacylglycerols. So, for the formation of a phospholipid, we need two compounds. One is a glycerol, another is fatty acid. So now we already know the formation of fatty acid. So let us understand how the glycerol is formed. So as you can see that here, glycerol is basically formed from one of the intermediates of glycolysis. So in glycolysis, we know that glucose is broken down in a stepwise manner. So when glucose gets broken down, one of the intermediates is DHAP. DHAP stands for dihydroxyacetone phosphate. So this DHAP can be converted into glycerol 3 phosphate by simple reduction reaction, or directly the cell converts glycerol into glycerol 3 phosphate. So first you need to know that there is formation of glycerol 3 phosphate in the formation of phospholipid, and then there will be attachment of a particular fatty acid to glycerol 3 phosphate, resulting in the formation of particular mono, di or triacylglycerol depending upon the number of fatty acids which are attached. So in this particular reaction, as you can see that this particular glycerol 3 phosphate now is basically getting connected with a particular fatty acid. Now remember that for the attachment of a particular fatty acid, there is a requirement of a coenzyme A, because coenzyme A is a transporter molecule, and these fatty acids are basically transported from the mitochondria into the cytoplasm. So directly the fatty acids cannot be transported and they cannot cross the membranes of mitochondria, and that's why they require a transporter molecule, that is coenzyme A. So first the fatty acid will get attached to a coenzyme A, as you can see here, and once a fatty acid has been attached to a coenzyme A molecule, then can get attached to the particular glycerol 3 phosphate. So now as you can see that in this particular picture, there are two fatty acids which are attached to the glycerol 3 phosphate, and this particular structure which is formed is known as a phospholipid, which is also called as phosphatidic acid, because there are two fatty acid chains and on the third carbon there is only a presence of single phosphate. Such a resultant product is called as glycerophospholipid, or you can also call it as a phosphatidic acid. Now this phosphatidic acid can be further converted into a triacylglycerol, that is the third carbon again can be attached with a third type of fatty acid chain. So if the third carbon is attached with the fatty acid chain, the net result will be a triacylglycerol. Or if there is a addition of a particular type of an amino acid on the third carbon, that is a particular head group is attached on the third carbon, then the net result will be a particular type of a membrane phospholipid. So different types of head groups can be attached to the third carbon, that is it can be serine, choline, ethanolamine, etc. So as you can see here that there is a particular head group which is attached to the third carbon. So as you can see that here the 1,2-diacylglycerol is getting converted into triacylglycerol by attachment of a third fatty acid on the third carbon, or here particularly on this side there you can see that there is an attachment of a particular type of head group, and there will be a different type of a phospholipid getting formed depending on the type of a head group which is attached. So now you have to remember that how a particular head group is attached to a third carbon. So for that, remember there comes a role of a particular intermediate, that is phosphoric acid. So in this particular picture you can see that phosphoric acid will condense both the OH groups. One OH group is of the head group and the other OH group is of diacylglycerol. So both the OH groups will get connected to the phosphoric acid, resulting in the formation of a bond which is nothing but a phosphodiester bond, and net result is going to be a particular formation of a phospholipid, a type of phospholipid, depending on which type of head group it has. So remember that phosphoric acid can help in connecting the OH group of head group and the OH group of diacylglycerol. Now in this particular diagram, you will understand that how exactly different types of phospholipids are formed. Now there was an important pathway which was discussed by the great scientist Eugene Kennedy in case of bacteria, that is E. coli. So Eugene Kennedy had explained two different strategies by which the phospholipids can be formed in the bacteria. Now see that one strategy is a particular diacylglycerol. As you can see, a particular diacylglycerol will get connected to this particular nucleotide, which is CDP. CDP stands for cytidine diphosphate. Now CDP is a type of a nucleotide. So if diacylglycerol gets attached to this CDP, the resultant structure is going to be CDP diacylglycerol. So once the CDP diacylglycerol is formed, then it can attach to a particular head group with the help of this OH group. So there will be a bond formed between the phosphate and the OH group. There will be removal of one CMP, that is cytosine monophosphate, and the resultant structure is going to be a particular phospholipid. So this is one strategy. That is, in this we saw that first there was formation of CDP diacylglycerol, and then CDP diacylglycerol was attached to the head group. The second strategy is the CDP, that is cytosine diphosphate, will first attach to the head group, and this particular entire structure will now attach to the OH group of 1,2-diacylglycerol, right? So try to understand this. One method is first 1,2-diacylglycerol will attach to CDP, resulting in the formation of CDP diacylglycerol, or CDP will first attach to head group and then it will attach to 1,2-diacylglycerol. In both the ways, what will happen is one CMP is going to be removed and finally there will be a formation of a particular type of phospholipid. So this strategy was explained to us by Eugene Kennedy in case of E. coli. So now in case of bacteria, what happens next is once CDP diacylglycerol is formed, then there can be addition of one more glycerol 3-phosphate. Once the glycerol 3-phosphate is added, one CMP will be removed and the net structure is going to be phosphatidyl glycerol 3-phosphate. On the other side, if there is addition of particular amino acid, that is serine, again one CMP will be removed, the resulting structure will be phosphatidyl serine. Now once phosphatidyl glycerol 3-phosphate is formed, from this side there will be removal of phosphate, the resulting structure is going to be phosphatidyl glycerol, or if from this particular phosphatidyl serine, if we remove one CO2 carbon dioxide, then the resulting structure is going to be formation of phosphatidyl ethanolamine. So there are different ways by which the cell can form different type of phospholipids in the cell, and as we just had discussed earlier that all these different types of phospholipids are required for the different types of cell signaling in the cell. Now you can see here that there is a formation of an important phospholipid called as cardiolipin, and how this cardiolipin is formed is majorly by the connecting two phosphatidyl glycerol. So here you can see this is one phosphatidyl glycerol, this is another phosphatidyl glycerol. So when two phosphatidyl glycerols combine, there is removal of a single glycerol and the net resultant structure is basically a type of phospholipid which is known as cardiolipin. So likewise there are different types of phospholipids which can be formed in the cell. Here you can see that there is addition of inositol again, which is a different type of a sugar which is added to the third carbon. So this inositol attaches to the phosphate group, resulting in the formation of phosphatidyl inositol. So all these are different types of membrane phospholipids which are formed in the cell. So whether it is a bacterial cell or eukaryotic cell, different types of membrane phospholipids can be formed in the cell. So I hope that you have understood the formation of different types of phospholipids in this particular video tutorial. If you have understood the concept, please like and share the video with your friends. If you have any doubts or suggestions, do mention in the comment section, and don't forget to subscribe the channel One Page Biology. See you all in the next video with some another biology related topic. Till that time, take care of yourself. Thank you so much. Bye. Hello everyone, how are you all? I hope you all are doing fine. In this video, we are going to discuss another very important topic called as cholesterol biosynthesis. So let us understand, this topic is important for all the different types of PhD entrance exams as well as CSIR NET Life Science examination, set exams, and this is part of even the uh, BSc biotechnology courses or MSc biotechnology courses in respective colleges. So let us understand this important topic, that is cholesterol biosynthesis. Now what is cholesterol? Cholesterol is basically a type of a lipid. It's an important compound because it has got different functions in our body, right from formation of steroid hormones, that is different types of steroid hormones like progesterone, estrogen, testosterone, etc. Whether it is the formation of different types of bile pigments, bile acids, or even in maintaining the fluidity of membrane. So cholesterol is very important. So how is exactly cholesterol formed? Let us understand. So cholesterol formation occurs in majorly 4 stages. What are these 4 stages? In the first stage, there is formation of a 6 carbon compound known as mevalonate. We will see how the mevalonate is formed. In the second stage, there will be formation of activated isoprene units. So this mevalonate will further get converted to activated forms of isoprene units. In the next stage, the isoprene units will condense together to form a long chain 30 carbon compound called as squalene, and finally this squalene is going to undergo cyclization, resulting in the formation of steroid rings, and what will formed as a compound called as cholesterol. So let us understand in details about how the cholesterol is formed. Now as we said, in the first step, a 6 carbon compound, that is mevalonate, is formed. So how mevalonate is formed? Mevalonate is formed with the help of acetyl CoA. So as we know that acetyl CoA has got only 2 carbons. So for the formation of mevalonate, there will be 3 acetyl CoAs which will be required. So in the first step, 2 acetyl CoAs will combine together to form a compound which is called as acetyl CoA. In the next step, this acetyl CoA will further add one more acetyl CoA with the help of enzyme HMG CoA synthase, and the net result will be the formation of a 6 carbon compound, that is beta hydroxy beta methyl glutryl CoA. So this is a very important compound called as HMG CoA. So HMG CoA, again let me tell you, the full form is beta hydroxy beta methyl glutryl CoA. So as you can see, this is a 6 carbon compound. Now in the next step, this beta hydroxy beta methyl glutryl CoA will undergo reduction, as well as there will be a removal of coenzyme A. So as you can see, on this particular carbon, there is addition of hydrogen resulting in the formation of CH2OH, and there is removal of this coenzyme A. So the net resultant structure is a 6 carbon compound which is mevalonate. So as we had said that the first stage in cholesterol formation is the formation of 6 carbon compound, that is mevalonate. So this mevalonate has been now formed. Now in the next step, this mevalonate will undergo 3 successive phosphorylation steps. So phosphorylation means, you all know that it is the addition of phosphates, and you all know that who donates the phosphate in a cell? It is nothing but ATP. So you will understand that now there are 3 phosphates which are going to be added by ATPs. So in the first step, Mavolonate will first undergo, in the first step, Mavolonate will get added with 1 phosphate, resulting in the formation of 5-phospho Mavolonate. Then there will be again second phosphate which will be added, resulting in the formation of 5-pyrophospho Mavolonate. In the next step, again there is addition of one more phosphate, but this time the phosphate is not getting added to the first carbon, it is getting added to the third carbon, and that's why the compound which is called, as you can see, this is the third carbon, so the phosphate is getting attached over here. So the compound which is formed is now 3-phospho 5-pyrophospho Mavolonate. So we saw that Mavolonate underwent 3 continuous phosphorylations with the help of 3 ATPs. This 3-phospho 5-pyrophospho Mavolonate will undergo removal of carbon dioxide, plus there will be removal of phosphate. So this carbon dioxide is going to be removed and one phosphate will be removed. So once the carbon dioxide is removed, there is a formation of a double bond over here. So the net resultant structure which is formed is nothing but delta raised to 3 isopentenyl pyrophosphate. So this is a very important compound, delta raised to 3 isopentenyl pyrophosphate, which will immediately convert into its isomer, which is dimethyl allyl pyrophosphate. So remember, delta raised to 3 isopentenyl pyrophosphate and dimethyl allyl pyrophosphate both are isomers of each other, and these are nothing but the activated isoprene units, because these isoprenes are going to be required ultimately for the formation of cholesterol. So once these activated isoprene units are formed, now what will happen? Again, these isoprene units are going to condense with each other. So one dimethyl allyl pyrophosphate will condense with another delta raised to 3 isopentenyl pyrophosphate, resulting in the formation of a compound which is geranyl pyrophosphate. Geranyl pyrophosphate will further undergo condensation with another delta raised to 3 isopentenyl pyrophosphate, resulting in the structure called as farnesyl pyrophosphate. So this farnesyl pyrophosphate is nothing but a 15 carbon chain compound. It's a long straight chain compound. So finally this farnesyl pyrophosphate, which is a 15 chain compound, will undergo condensation. So there will be two farnesyl pyrophosphates, that is 15 chain and plus 15 chain, both will join together, resulting in the formation of a 30 chain compound, which is nothing but squalene. So when two farnesyl pyrophosphates join together, there will be removal of these two pyrophosphate groups, and with the help of hydrogen which is donated by NADPH, both the farnesyl chains will be joined together, and the net result is going to be a straight chain compound which is called as squalene. Now the important step is, this squalene will now undergo addition of oxygen. So remember, the addition of oxygen occurs with the help of the enzyme called as squalene monoxygenase. So as you can see, the oxygen is added. So the oxygen gets added on the third carbon, and once the oxygen has been added with the help of monoxygenase, there will be immediate reduction reaction which is carried out by the help of NADPH. So NADPH will donate the hydrogen and the oxygen will be converted to the OH group. So the net result will be the formation of a OH group, as you can see. And also you need to remember that once the oxygen has been added, there is a characteristic placement of this double bond. So what we should know is the cholesterol structure has got these double bonds which are present, and because of the presence of these double bonds, there is a characterization, there is a particular cyclization of these structures. So ultimately this squalene 2,3 epoxide will be converted to stigmasterol in case of plants, or it will be converted to lanosterol in animals, or ergosterol in fungi. And finally this lanosterol will undergo 20 different reactions, 20 different reactions in which there will be addition of methyl groups, And finally, the lanosterol will be converted to cholesterol in case of animals. So this particular pathway was found by the great scientists, that is Konrad Bloch, Fyodor Lenin, John Conforth, and George Popjak. So these were the great scientists who elucidated this particular pathway of cholesterol biosynthesis. So I hope you have understood this particular pathway of cholesterol biosynthesis. If you have understood this pathway, please like the video, share it with your friends, and do subscribe to One Page Biology. See you all in the next video with some other biology related concept. Take care of yourself. Thank you so much. Bye bye.